So unlike Joe Berger, my first slides is always the same boring picture of the building where I do my research. I'll try to think of something more pleasing for the eyes for the next workshop. So as you've heard in other talks before, polyomaviruses, including JC virus, have a capsid formed by 72 pentamers of the VP1 protein. Here, this is on electron microscopy. This is a schematic rendition where you have these pentamers of VP1 that are associated inside to either the VP2 or VP3 capsid protein that surround a single copy of double-stranded viral DNA. And this is a schematic rendition of the genome of polyomaviruses, including JC virus. You heard from Jean Major's talk this morning. It's a small circular DNA of 5 kb, like a little plasmid, where you have a very active regulatory region, which is hypervariable and usually a very conserved coding region formed by the small t and large t regulatory protein on one side, the early genes, and mainly the capsid gene VP1, VP2, and VP3 on the late side of the virus, and the agnoprotein, which is also a regulatory protein whose function has not entirely been elucidated. So this is an excellent paper from Jean Majors already from 2001 that depicts in a compass form all the different forms of non-coding regulatory region in various compartments. If you heard about that before, this is a region which is hypervariable. There is no single type of variation. Every PML patient has a regulatory region that's different from the next, and it's felt to contain determinants of neurotropism and neurovirulence. And the common, wisdom that we get from these talks and what we write also in review articles is that archetype regulatory region is found in the kidney and the urine of everyone, regardless if you're healthy, HIV infected, with or without PML, you'll have archetype mainly in the kidney and urine, and the rearrange or tandem repeat form are found predominantly in the spinal fluid and the brain of the patients who have already developed PML. And this is good if you like to see the reality in black and white. It's not always the case, and if you look hard enough and doing a lot of cloning and sequencing of multiple clones of the regulatory region in various patient specimen and sample, you will find that sometimes you find tandem repeat forms in extraneural site of HIV-infected patient with PML, for example, and sometimes you also find the archetype form of the regulatory region in the brain of patients who have developed PML. And this has been shown by a number of other groups, and obviously when you take one sample at one given point in time, let's say in the blood or in the bone marrow, you don't know if it's a virus that's necessarily originated from there or went first to the brain and then back to the bone marrow at the time where you pick up the virus. Now, we discussed already that the JC virus coding region is extremely conserved, and actually it's been used by epidemiologists to follow the migration of ancient population, which is very interesting. Globally, you have to remember one thing, is that there is one serotype of the virus and 3 major genotypes with other subtypes, which are based on point mutation of the VP1 genes. And between the different types, you have between 1 and 4% of nucleotide mutation that lead to a change of few amino acid. And genotypes have been associated with the geographic origin of the patients. For example, type 1, mainly European patient, type 2 Asian Native Americans, type 3 Africa, and so on. And there have been few molecular epidemiological studies that have been done on large samples of various samples of virus from a given patient, for example. But this study showed that there was a limited number of synonymous substitution per site per year, which indicates that usually you don't have polymorphism in a given patient, and you expect that in a single person is infected by a single strain. Now, over the years, people have tried to associate different genotype with disease activity. And for example, way back when in 1997, these researchers looked at that issue and found that genotype 2B was more frequent PML lesions than in the urine of healthy people. But they were testing they were looking at the virus from PML patient in the brain and compared it with the urine of healthy people, not with the urine of the same PML patients. And so now I'm going to summarize 3 papers that have been published by a researcher from Biogen IDEC, sometimes in collaboration with other researchers like Paola Cinque in Milan, where they looked at isolates from PML in the CNS that were compared with the urine sample, again from healthy controls, and all these data were deposited in GenBank, and they were just analyzing all these data that were already generated by others. This was published in 2009, and interestingly, they found no difference in genotype and disease activity, but they noted that half of the PML isolates had at least one amino acid mutation in the VP1 gene compared to urine of healthy controls, and noted in position 55, 60, 265, 267, and 269. When modeling JCV VP1 tetrasaccharide complex, suggested that these sites, amino acid sites, were implicated with the binding to JCV receptor, and when they made a viral-like particle containing some of this mutation, but not all, for example, these 2 variants failed to hemagglutinate type O erythrocyte. And hemagglutination capability of JC virus is well known for many years, and it's been used actually in an hemagglutination inhibition assay to detect who was seropositive for JC virus before the new ELISA assay were available. And the conclusion of this paper was that lesser binding of JC virus in the periphery may promote its entry in the brain. So when you look at this data, I think it's interesting. The caveats that we need to discuss today is that most published sequence of JC virus in the literature are obtained from analysis of a single clone or of direct sequencing of PCR product. And when you do that, you're going to get the major species that is available in that compartment. You don't have a good idea of the variability that is present. In addition, they compared PML isolates with the urine from the healthy controls, not with the urine sample from the same PML patients. And initially, the structural modeling was based on murine polyomavirus, then was adapted and retrofitted to a JC virus when the crystal structure of JC virus VP1 became available. So this same group of researchers, I think in collaboration with Paola Cinque in Milan, then looked at pair urine, plasma, and CSF sample from PML patients, and that was published this year in Journal of Infectious Diseases. And again, they looked at the VP1 gene, which was analyzed by PCR, followed either by direct sequencing or by cloning and sequencing. And they had 17 cases, but in fact, only 7 of these cases had JCV that was cloned from both urine and the CSF. And you can see in 2 out of 7 cases, there was a mutation at the 122 amino acid that was present in CSF, not in the urine. In 3 of these cases, mutation at position 269, 1 out of these 7 position 55, and in 1 out of 7 cases, there was no change between the urine and the CSF sample. So interestingly also, most CSF samples had identical sequences compared to the plasma sample. And when they constructed viral-like particle, they saw that some of these mutations, but not all of the mutation, had decreased binding to gangliosides, failed to hemagglutinate the type O erythrocytes, and had decreased binding to kidney cells and lymphocytes, but not to astroglial cells, which was, by the way, not the case of this mutation at position 269. And then they saw also that the binding was not affected by neuraminidase treatment of astroglial cells, which meant that this was sialic acid independent. So the conclusion of this paper, which I think is interesting, is that the mutation leading to lack of sialic acid binding resulted potentially in escape from trapping of the virus in the periphery and leading to the access of the virus to the brain, but that did not affect binding to astroglial cells, which are the preferred target of JC virus. Again, a critical reading of this paper is they compared the CNS isolate with what they call the wild-type isolate, which is MAD1. But in fact, MAD1 is also a CNS isolate. It was initially derived from the brain of a patient with PML. So wild-type is actually a CNS isolate. As we discussed, there's a limited number of paired urine and CSF sample, 7 as a total, and out of these 7 there were many mutations which seem to have different properties in different assays. Interestingly also, the PBMC, peripheral blood mononuclear cell, were not tested, only virus from plasma, and I think this would be important to look at PBMC since this cell may be also instrumental in bringing the virus to the brain. And there was no discussion of statistical significance, especially when multiple comparisons were established. And finally, the authors discussed that also in their papers. We're not really sure if mutations are occurring before or after the virus entry into the central nervous system. So the last paper by this group, also appearing in the same issue of Journal of Infectious Disease, looked then at paired urine, plasma, and CSF sample from 17 natalizumab-treated patients who developed PML. And again, they looked at the regulatory region, the VP1 gene, and did sometimes full-length sequencing analysis by PCR, followed either by direct sequencing or cloning and sequencing. They had 17 cases, but only 5 with both urine and CSF sample. And in these 5 cases, they found one time each these mutations at position 55, 61, 66, 267, 269 in the CSF, but not in the urine of the same patients. And only 4 out of 6 paired CSF samples had the same mutation, did not find any VP1 mutation in the 6 urine samples. I think what was very interesting is that they were able to do a full-length genome analysis in 6 patients and analyze 2 to 45 clones per patient, which is a very big undertaking, and found 1 genotype per patient, but again, no association between genotype and PML, which confirms their earlier observation. There's a sentence that we need to clarify, that JCV in the CSF was more heterogeneous than in the urine of the same patient, and we could discuss what it means. The regulatory region rearrangement were independent from the VP1 mutation, but these mutations occur always concomitantly with regulatory region rearrangement. So their conclusion for that paper is that JCV natalizumab PML was similar to other PML cases, that the regulatory region rearrangement were more common and appear to precede VP1 mutation. There was no specific association of this rearrangement with a typical one or the other VP1 mutation, and that these rearrangements were similar in the plasma and the CSF. And they postulated that you had a common asymptomatic infection of the archetype in the kidney, with then rare occurrence of changing the regulatory region and VP1, leading to CNS infection. Again, when you try to get a slightly critical reading of this manuscript, I think it was a good start. Again, we have a limited number of paired urine and CSF samples, obviously in this limited population, where we have multiple VP1 mutations. All samples were collected at the time of PML diagnosis, obviously, and therefore it's unclear whether the VP1 mutations occur prior to the clinical manifestation of PML. The exact route of primary infection has not been determined, and it is unclear if the primary infection occurring in all of us occurs with archetype type regulatory region or already reorient regulatory region plus or minus VP1 mutation. Now, what are the solutions to these problems? Well, I think it would be worthwhile to perform cloning and sequencing of multiple full-length JCV clone per patient. And the full-length PCR amplification and sequencing of multiple clones of JCV virus in a single patient at multiple time points would be needed to evaluate the genetic evolution and intra-patient diversity, which is something that's never been done before. I think it would be interesting to discuss about the data already obtained from multiple clone of the CSF from MS-PML patient and what it means that the genetic heterogeneity was higher than in the urine sample from the same patients. I think it would be a worthwhile undertaking to determine the JCV VP1 sequence in a larger number of urine, plasma, PBMC, and CSF of PML patients over time, regardless if they had natalizumab or not, or if they are HIV infected or not. I think it would be also interesting to look at the urine, plasma, and PBMC in immunosuppressed patients without PML over time and see the genetic diversity in this patient population, and also look at the genetic variability in urine of natalizumab-treated MS patients over time in these cohort studies. And finally, longitudinal studies with deep sequencing of JCV virus in different compartments in natalizumab-treated patients may also shed light on these questions. Now, since we're talking about a change in the sequence of JCV virus, just want to finish up and alerting you to two novel syndromes caused by JCV variants, also coding region variants, which, as Joe Berger just told you, highlight the fact that JCV virus can not only infect glial cells, but also neurons. And E.P. Richardson, who described PML in 1958, already saw that they were in the PML patients' brain sample, there were area of focal cell loss in the internal granule layer of the cerebellum in 5% of these. And Joe Berger and others in a publication in 1998 showed that AIDS patients had a cerebellar atrophy that was otherwise unexplained. And we had the occasion to see an HIV-infected patient in PML who had regular lesion of PML in the cerebellar white matter with positive JCV virus PCR, also had cerebellar atrophy and cerebellar syndrome, which was not explained because there was no lesion of PML in this area. And just to remind you, if you don't look at this every day, the cerebellum has white matter, then a granule cell layer, which is formed of granule cell neuron, interneurons, and then you have Purkinje cells and a molecular layer on the surface. And you would expect to find PML in this area of the white matter, but not in this gray matter area formed mainly of neuron. But what we saw is that there was no lesion of PML in the white matter, but there were area of cell loss, as you can see here in the granule cell layer. And in the periphery of this area of cell loss, you had neurons that stained positively for polyomavirus antigen. And that was reported in my lab by Renaud Dupasquier in 2003. And we were able to do electron microscopy in this sample and show that the granule cell neuron had mature JCV virus in their nuclei. And to make a long story short, we were able to sequence the entire virus and found a 10 base pair out-of-frame deletion in the JCV VP1 protein, resulting in a change of 13 amino acids at the C-terminus. And the C-terminus is the little hook that keeps the pentamer of JCV virus together. And this is the area that is mutated, which is, by the way, very different than the area that was reported by the Biogen IDEC group in the previous papers. And nowadays, there have been 10 case reports of JCV Granule Cell Neuronopathy published in the world, in the United States, East Coast, West Coast, in Brazil, in Chile, in Australia, in South Korea, and hopefully we can expect more to be diagnosed in other places in the world. Now, another novel syndrome, and I'm telling you this because that may be also important and useful in natalizumab-treated patients, that we observed recently, 74-year-old HIV-negative lady with gray matter lesion and encephalopathy. The lesions were in the cortex, in the gray matter, exactly the opposite than what you would expect with PML. To make a long story short, again, this patient unfortunately passed away. We're able to do an autopsy restricted to the brain, and using a variety of neuronal marker, MAP2 or NUN, and either T antigen or VP1 protein marker for JC virus, you can see that the viral protein are expressed inside of the neurons, both the regulatory protein and the capsid protein are expressed inside of the neurons, and we're able to do electron microscopy showing that you had mature viral particle in the nuclei of this cortical pyramidal neuron, and that form a new entity that we call JCV encephalopathy or JCVE. So this is our vision of the world, and where we have classic PML that we discussed at this meeting, where sometimes you can find single amino acid mutation in the VP1 protein, but should not discount other manifestations of JC virus, such as JC virus granule cell neuronopathy, where you have alteration in the VP1C terminus and lytic infection of granule cell neurons, and finally JCV encephalopathy, which you have cortical lesion in the gray matter, and preliminary data indicates that you have a novel mutation in the agnoprotein gene in these patients. And I want to acknowledge again my collaborators and people who help us with this research. Thank you very much.